Good evening, students. So, in this session, we are going to study the molecular structure and functions of ribosomes and lysosomes. Okay, so coming to ribosomes, ribosomes are the granular structures first observed under the electron microscope. Okay, so as a dense particles by George Pallade. Okay, by name George Pallade. So in 1953. Okay, so in 1953. So the ribosome word is derived ribo from ribonucleic acid and somes. Okay, so it is derived by soma, which means body. So they are composed of ribonucleic acid and proteins and are not surrounded by any membrane. They are not surrounded by any membrane. Okay, so they are tiny spheroidal dense particles. They are circular in shape and having a 150 to 200 angstroms in diameters. So that are primarily found in most prokaryote and eukaryotes. That means ribosomes are found in every cell, and they are the main sites of a protein synthesis, and they have a structure approximately equal to the RNA. That means amount of RNA is directly proportional to the ribosomes also. Okay, and they also contains equal amount, or it may be a somewhat differentiation in the amounts of RNA and proteins which they contain, and also they have a scaffold. That means they are keeping a height. That means okay, ordered interaction of the numerous molecules involved in protein synthesis. Okay, the and ribosomes occur in cells, that is, as I said, both in prokaryotic and eukaryotic cells. And in prokaryotic cells, the ribosomes often occurly freely in the cytoplasm. Okay, they are occurring freely in cytoplasm, as you are seeing in this picture. And these ribosomes are the site of protein synthesis. In eukaryotes, yeah, the ribosomes either occur freely or they are that is freely in a cytoplasm or attached to the endoplasmic reticulums. That is by the appearance of these ribosomes, and the endoplasmic reticulum look a rough surface. So it is called as rough endoplasmic reticulum. Okay, and the location of the ribosomes in a cell determines what kind of a protein it makes. That means if the ribosomes are freely in a floating in a cytoplasm, okay, they are synthesized in proteins which are utilized by itself. That is utilized by the cell only. Okay, if the ribosomes which are producing uh, proteins which are attached to endoplasmic reticulum. Okay, so they are there is a rough endoplasmic reticulum. So these ribosomes which they secrete proteins, they are used by inside the cell and also outside of the cell. Okay, and the number of ribosomes in a cell is depending upon the activity of the cell. That is a type of cell which they involve in a biochemical reactions. Okay, on average in a mammalian cell, you can find about 10 million of a ribosome. So this is a structure. There is a larger subunit and a smaller subunit. Okay, and larger subunit is having a P site, mRNA binding site, E site. So it is clubly called as APE site. Okay, so these are the tRNA. Okay, and this is the small subunit, and these dots are the mRNA. Okay, where this process is explaining the protein synthesis, or we can say the translation processes. Okay, in the eukaryotic ribosomes, they are containing the 80s, while prokaryotic ribosomes are containing 70s. Okay, 70s. So that is a subunit. So it is said that the density, what is the density? That means ribosomes are composed of two subunits, okay, that come together to translate messenger RNA. Translate means the codons are on the mRNA. So according to that, the tRNA sends a message as anticodons. That means the translation from codon to anticodon. By that, the polypeptide synthesis takes place. Okay, by that the proteins are synthesizing. So the volume is measured in Swedberg units. As Swedberg means it's a name of the scientist. Okay, yes, Swedberg units, a unit of density corresponding to relative rate of sedimentation during ultra high speed centrifugation. Okay, so the greater the S value, the more dense the particles. That means it's depending upon the S. Okay, S means it is Swedberg as a site. Okay, so this is centrifugation process, centrifugation unit. This is normal centrifugation and it is ultra centrifugation. Okay, so units as RPM is denoted. RPM means rounds per minute. Okay, so here ultra centrifugation you can get 22,000 RPM. Okay, this is the socket which is revolving around. Okay, it is denoted by this RPM, rounds per unit. Okay, if 22,000 RPM, okay, it is a test tubes or open of tubes which are kept here. Okay, so while revolving at a speed of 22,000 rounds per minute, then these organelles can be settled down. Okay, this is this centrifugation process. Okay, and this is completely depending upon the sedimentation or the density of the molecules. Okay, so as I said, the prokaryotes having a total 70s subunits and eukaryotic is having a 80s subunit. So in a 30s subunits, it is a small unit. In eukaryotes, it is a 40s subunit, which is comprising of 18s and 33 proteins. And 30s subunit in a prokaryotes is having a 16s and 21 proteins are there. In a larger unit in the prokaryote, 50s subunit is there and 60s subunit, which is comprising of 28s RNA, 5.8s RNA, 5s RNA, and also 50 proteins. That's combinedly we can say the 80s RNA, 80s ribosome can be seen in a eukaryote and 70s subunit can be seen in a prokaryotes. Okay, the structure as we discussed earlier, it is a made up of mainly RNA and proteins. Okay, therefore it is also called as ribonucleoprotein. Okay, and 37 to 62% of RNA is composed of RNA. Okay, up to 62%. So rest will be proteins. And as we seen in last slide, each ribosome is divided into two subunits, smaller and a larger. Okay, and smaller units which binds a larger subunit and the mRNA pattern, and larger subunit which binds to the tRNA and amino acid, and then smaller subunit. Okay, so prokaryotes have a 70s ribosomes, okay, which is having a 30s and 50s subunits, and the small subunit 16s having a 1540 nucleotide base pairs. Okay, it contains 1540 nucleotides and it bond with the 21 proteins. And the larger subunit is composed of a 5s RNA, which is having a 120 nucleotides, 23s RNA subunit having a 2900 nucleotides, and also comprising 31 proteins. And in a eukaryotes, the 80s ribosomes, which is comprising of a 40s and 60s subunits. Okay, 40s subunits, it is in a prolate ellipsoid in a shape and consists of a 18s ribosomal RNA, or we can say rRNA. Okay, and also 30 proteins. That 30 proteins are named as a S1, S2, S3 up to S30. That is a smaller subunit proteins. And the larger 60s ribosome subunits is in round in shape and contains a channel through which growing polypeptide chain makes it exit. It is an entry and exit valves will be there in a 60s ribosomal RNA for a tRNA. Okay, and it consists of three types of rRNA: 28s and 5.8s and 5s, and also 40 proteins. So these 40 proteins named as a L1, L2, L3 up to L40. So as a larger subunit proteins. Okay, and the difference between the ribosomes of this and eukaryotes, that means you use to create antibiotics that can destroy bacterial infection without harming human cells. Okay, and the ribosomes, since in a chloroplasts and the mitochondria, they are having also 70s particles. And the ribosomes share a core structure which is similar to all prokaryotic and eukaryotic cell. That is the contents of ribosomes. Okay, and the two subunits work together on a single place. Okay, so on a mRNA to translate its codons to anticodons. By that, the polypeptide chain is happening, and by that, protein synthesis takes place. Okay, so because as they are formed from two subunits non-equal size, they are slightly longer in axis than in diameter. So during protein synthesis, when multiple ribosomes are attached, they are called as polysomes. Okay, these ribosomes, they exist actually temporary. After protein synthesis, after polypeptide chain formation, the two subunits separate and they can be reused, and also they can be broken up. Okay, so as I said, this is the structure of ribosomes, that is a larger subunit which is having A, P, E sites, exit site, and this is the A site where the tRNA binds amino acyl tRNA. Okay, tRNA with the amino acid called as amino acyl tRNA. This is the site, and P site is a peptidyl tRNA where it is attaching to the another polypeptide. Okay, and E is the exit site where tRNA separates from the amino acid and they are existing. Okay, and they exiting from this unit. So this is a smaller subunit, this is larger subunits, and this is the mRNA. And the functions of mRNA, as the major function includes protein synthesis, or we can say translation. Okay, this is mRNA, and these are larger and smaller subunits. So here the sites are their APE sites. So here amino acid, it is the tRNA with the amino acid. Okay, it is coming inside the larger subunits, and the amino acid which is attached to previous tRNA is tend to attach with this amino acid, and they make a bond. Okay, so that bond and the making a chain of amino acids, the process are called as peptidyl transfer and the peptidyl hydrolysis. This process also takes place while in a translation. Okay, so then coming to next, lysosomes. Okay, lysosomes, they are the structures which are membrane bound. Okay, and they having hydrolytic enzymes which are responsible mainly for intracellular and extracellular digestion. Okay, the word that is lysosome is made up of two words. Actually, lysis, it's meaning breakdowning, and also you can say it is a killing, and soma means it's a body. Soma means body. Okay, so it is an important cell organelle responsible for the, as we said, internal and extracellular breakdown of substances. It may be lower molecules or higher molecules. So they are more commonly found in animal cells, but on some lower plant groups, that is fungi, they are there on a plasma membrane. Okay, and the lysosomes occur freely in cytoplasm. Okay, they are present freely in cytoplasm in animals, found in almost all cells except in a rbc. Okay, rbc is not having these structures, and they are found in most abundant numbers in cells related to, that is enzymatic reactions of the particular cells, that is the liver cells, pancreatic cells, kidney cells, spleen cells, leukocytes, macrophages, etc. Okay, so this is structure of lysosome. So it is having a membrane. Okay, and inside that the enzymes are there, just looking as a granules. Okay, so this is the diagram representation of a lysosome, and the structure includes. So they are without any characteristics shape. That is, they are present as a pleomorphic. Okay, no having a particular shape. and they are mostly globular and also granular appearance. Okay, and the size of this lysosome is in between 0.2 to 0.5 micrometer, and they are surrounded by a single lipoprotein membrane. Okay, that is a unique combinations which cannot be seen in other cells, other cell organelles. Okay, and these proteins are two types of proteins. Okay, they are both are glycosylated proteins only, and they are two types, lamp and limp. Okay, lamp means lysosomal associated membrane proteins, and limp means lysosomal integral membrane proteins. Okay, so both these proteins form a coat on the inner surface of the membrane. Okay, inner surface of the membrane, and they protect the membrane by the action of this hydrolytic enzymatic mixture. Okay, these enzymatic mixture can destroy this membrane only. Okay, so to overcome this, these proteins are coated. That is a lamp and limps. Okay, the lysosome membrane, it is also having a hydrogen proton form. That means they are maintaining the pH level, and inside the membrane, the organelle contains the enzymes in the crystalline form. Okay, so how these lysosomes are manufacturing means, okay, when the endoplasmic reticulum undergo protein synthesis and also glycosylation, okay, by that these vesicles are transferred to Golgi complex in a cis, okay, cis way. Okay, these are the cis membranes and medial membranes and trans membranes. The Golgi complex and three set of membranes. Okay, the cis membrane is towards the endoplasmic reticulum. Okay, when these vesicles enter into the cis membrane, okay, then phosphorylation of M6P taking place. M6P means mannose 6 phosphate. Okay, so when phosphorylation of this sugar taking place, okay, so and from transmembrane, okay, so these lysosomes are converting as a transport vesicles, then they get a maturation and the complete lysosome formation taking place. And there are a set of enzymes. Okay, that means lysosome contains a set of enzymes. They are equally called as hydrolysis. Okay, so they can tend to break the hydrogen bonds, so they are called as hydrolysis. Okay, so there are plenty of enzymes. Okay, naming it is a 40 or more than different varieties of enzymes are there. Okay, so some are listed here. That is a proteases, which can digest proteins, lipase digest lipids, amylase digest carbohydrates, nuclease digest nucleic acids, and phosphoric acid monoesterases. Okay, so these collectively called as hydrolysis, they can cleavage these substrates, okay, by the addition of especially water molecules. Okay, and the most of the lysosomal enzymes, they are in an acidic medium. Okay, so pH is having up to 4.6. Okay, so slightly acidic in a nature. Okay, then types of lysosomes are there. Okay, there are two types of lysosomes: the primary lysosomes and secondary lysosomes. Okay, the lysosomes which are freshly formulated by the endoplasmic reticulum and Golgi complex, they are primary lysosomes, and they are looking just like a storage of granules. Okay, the granules are nothing but an enzymes. Okay, and the secondary lysosomes, they are fusion with the phagosomes. Okay, phagosomes means after a phagocytosis, then the vesicles are forming. There is when the cell undergo the phagocytosis, that engulfing the foreign particles or food particles. Okay, they are called as phagosomes. Okay, these phagosomes combine with the primary lysosomes, they are forming the secondary lysosomes. Okay, they are also called as autophagosomes. They are called as autophagosomes, and they digest the complete part, and the exocytosis taking place there. The wastes are released from the cell, and the functions of these lysosomes majorly two functions, that is intracellular digestion and autolytic action. Okay, so to digest the food, okay, to digest the food, the lysosome membrane fuses with the membrane of a food vacuole, as we seen in last slide, food vacuole, And squids means the enzymes are uh, transferring from these lysosomes to food vacuoles. Okay, lysosomes, the enzymes which are present in lysosomes, they are squirted to, or they are transferred to these food vacuoles. Then the digested food then diffuses. Okay, then they can be digested and it diffuses through the vacuum membrane and enters into the cell, and that can be used for the energy production and also growth. And then autolytic action. So these cell organelles that need to be get ridden, that is apoptosis taking place. Apoptosis means the death of a cell. Okay, when cell undergo age, when a lifespan is ending, then the cell undergo death. That is a programmed cell death. Okay, so this is done by these lysosomes, okay, or these vesicles. That's a uh, condition called autophagosome. Auto means it's a self, and phagosome means the digesting of the internal cellular membranes or organelles. Okay, so these autophagosomes are then they later destroyed by the same lysosome enzymes. Okay, in the process, some other process which these lysosomes are included means uh, some are there, the heterophagy. Okay, heterophagy is also called as the endocytosis, engulfing. Okay, as you said, phagocytosis, that is nothing but a heterophagy only. Okay, so it is taking into the cell exogenous materials. Okay, that may be phagocytosis or pinocytosis, or pinocytosis means the engulfing of water molecules. Okay, and by that digestion taking place. And then the autophagy, the normal physiological processes. Okay, you can see autophagosomes. Okay, when the lysosome attack the food vacuoles, then the enzymes release into the food vacuole, then the digestion takes place. That's autophagosomes. And the extracellular digestion you can see on a saprophytic fungi, which is depending on the dead and decay materials for the degradation. And another process is there, that is autolysis. Autolysis means it is a apoptosis. You can see this picture, okay, where the tail is degenerating. Okay, tail is degenerating in a metamorphosis of amphibians, okay, frogs. And then in a fertilization processes, acrosome of a sperm contains, okay, many uh, these lysosomic enzymes which release the enzymes and they digest the egg surface so that the genetic material enters into the egg membrane. Okay, and as a janitor of the cell, janitor means these lysosomes are the cleaning processors. Okay, they clean the junk materials which are said as a waste, okay, which are accumulated in the cell, so that it can prevent the diseases also. And also these lysosomes are called as suicide bags, okay, or we can say programmed cell death, okay, by the action of their lytic engines. Here you can see when death stimulus is from nucleus, if arised from nucleus. Okay, so by that lysosomes releases cathepsin, and mitochondria releases autophagenic factors. Okay, these autophagenic factors influence lysosomes, and these cathepsins influence mitochondria, so that the caspase cascade formation taking place. Cascade means it's a group of enzymatic actions taking place, by that cell undergo death or a suicide. So for that purpose, these lysosomes are called suicide bags. And these are the clubbing of all the functions of lysosomes, okay, which can have glands, that is a positioning motility, and it's undergo gene regulations. It is helping for metabolic signaling, apoptotic cell death, plasma membrane repair, tumor invasion and metastasis, cell migration, cell adhesion, detoxification. Okay, so these are all the things which can be done by the lysosomes. Okay, so this is all about the molecular structure and functions of ribosomes and also ribosomes. Thank you.